Thanks, Jake. I'm glad to hear that you only found one error. That's encouraging. um, it is a huge honor to be able to give this talk, and my response to honors of this magnitude are to always think to myself, well, maybe someday in the future, if I keep on trying hard enough, eventually I'll be able to actually earn it. So that's the sort of attitude that I launched myself in with this. This was an enormously difficult talk to prepare because it sort of goes all over the map. I wanted to cover a fairly conceptual problem and tried to link it up with Neil's work, and we'll see if I succeeded. I'm afraid it's sort of a normal length seminar, so if people get tired, time is sort of moving on, so stomp your feet and I'll kind of speed up. How about that? So this is a picture of the retina of an individual with retinitis pigmentosa, and believe it or not, we're actually going to wrap the story up with this at the end, so pay attention and you'll hear a story about retinitis pigmentosa at the end. So I like this picture of Neil. It shows him hard at work. He relates the story in this book, which I did actually reread for this lecture. If any graduate student in the program who hasn't read it yet, it should be required reading. It actually is enormously readable for the historical perspective of what it was like to be a human geneticist in the early days and to sort of see the magnitude of discoveries that were yet to be made. So in his history, he actually did start with Drosophila, like all great geneticists. He worked with Kurt Stern at the University of Rochester. His thesis work was relatively unnoticed, I guess I would say. It wasn't terribly breakthrough. It didn't seem like he really fell in love with Drosophila like some people did. And he realized that he felt that the work in Drosophila was sort of nearing its completion, that there wouldn't be too much more to be discovered with flies. Of course, this preceded several Nobel Prizes, including the discovery of the innate immune system, the discovery of pattern formation in embryos, and so on and so forth. However, we'll leave that. People have felt that about Drosophila many times in the past, and it will come back again. So anyway, he wanted to apply the rigor of fly genetics to human genetics. And he was inspired by Salvador Luria, who wanted to apply this sort of rigor of physics to biology by entering into the study of viral genetics. And this was his idea of getting into human genetics. And he realized in order to do anything in human genetics, it really would be wise to understand human biology much better the way a physician could. And so still at the University of Rochester, he was able to work on his degree in medicine, which was granted in 1945. For his residency, actually, at Rochester, he worked on thalassemia and sickle cell. Sickle cell at that time we knew was a blood disorder. The sickling phenomenon was clear. Individuals who had sickling disease were clearly ran in families, but it actually wasn't even particularly clear that heterozygotes had a sickle cell trait and were sort of simply intermediate in phenotype as a result of their heterozygosity. And that picture of him running the gel actually was recounted in the book of him telling the story of Linus Pauling running the first electrophoretic gels, separating the normal from the beta S globin forms and realizing that the carriers of the sickle cell disease were in fact heterozygotes. Anyway, in 1947, he was named famously for to be the director of the field studies for the National Research Council's Atomic Bomb Casualty Commission, went to Hiroshima and Nagasaki and did terrific epidemiological work trying to study what were the consequences of the exposure to radiation of that population. And the upshot of that story, as everybody knows, is that the initial reports were actually surprisingly small effect. That is, the relationship human doubling dose for mutation was remarkably close to the lethal dose. And so individuals, if they weren't killed, the manifestation of mutation was actually not that obvious with the relatively crude ways that they could identify those effects at the time. And this earned him, some people really weren't so happy with that result. The obviousness of the terrible consequences of the nuclear bomb were painfully obvious to everybody, so surely there would be a genetic consequence as well. And it was just difficult to detect with the technologies available. The whole thing resurrected again in the 70s. You may recall that using molecular techniques at that time, electrophoresis and looking at chromosome abnormalities and so forth, many additional defects were found. And of course, the sea of cancers that appeared, the epidemiology of that was progressed greatly. Anyway, along the way, sort of just after that time, he was still a practicing clinical doctor. He became discouraged by clinical medicine, and this began his sort of this dichotomy of this, should his attention be paid on bettering the sort of population of humans, the sort of gene pool kind of physician, versus being just a custodian of death as the clinicians were in his mind. So he had this sort of falling out with the importance of that one-on-one contact that a physician has with the patients, and felt his greater impact could be at this other level altogether. Anyway, to get to that other level, we saw that genetics was really the ticket, that by studying genetics, he could attend to the primary cause of disease rather than just attending to symptoms. And so this gives rise to sort of a thread that runs through the book and runs through this talk a little bit, which is this sort of dichotomies of the ways that you see the world. So this one is this sort of idea of understanding things at the level of the sort of epidemiology, of population genetics, of the sort of gene pool, and what are the processes that are involved in the health of that, versus the one-on-one contact with patients and sort of attending to their difficulties. A sort of similar parallel is between thinking about problems at this epidemiological level, statistical descriptions of things, compared to the digging down and getting at the underlying mechanism. What is the gene? What are the molecular basis of the defects, and how do they work? And I'm setting this up as though these things are in tension, and the argument is going to be that eventually these things ought to come together at the end. One other tension that we see, and this actually you see within the National Human Genome Research Institute, is there are programs specifically designed for going after the genetics of complex traits, complex diseases, common diseases, and other programs designed at getting after Mendelian defects, so things that segregate in families like Mendelian disorders. And we go about the sequencing and the design of those studies completely sort of separately at this point. And this makes sense because of the relative frequencies and the ways that we go about them, and yet there are attributes of them that sort of intersect. So to what extent are complex traits determined by many, many different rare variants of relatively large effect versus common variants of small effect, and so forth. So that also gets into this mix of the way we think about these problems at these different levels. And now we're presented with this sort of mandate to think about precision medicine. The whole idea of precision medicine is to use all of this information up here to eventually feed back at this level. So how do we use all the statistics, all the sort of mechanism that we understand to actually feed back to making predictions at the clinical level? And hence the title of my talk, Getting Back to Prediction as being a fairly primary goal in genetics, in human genetics in particular. And yet it's one that we sort of set aside because it's so difficult, and we'll see why that is. So just to continue, and this will be the end, I think, of the discussion about Neil. So we mentioned his work on thalassemia, sickle cell. We mentioned his work on Hiroshima and Nagasaki. In doing that work, so it was basically epidemiological, collecting the information from families that were at different distances from the hypocenter of the blast and so forth. He understood that there's wide range of variability in the degree of consanguinity in those Japanese families. And this had enormous impact on the apparent familiality of the disorders that the family showed, and then the overall health of those individuals. So consanguinity became a focus of his, and he published several papers on that. We'll get back to that. I'll actually give a small vignette of work that we've done that relates to that work. He also, of course, did work in South America on the Yanomami Indians, the work on private polymorphism, the understanding of rare variants being highly localized geographically in human populations resonates today through Laurent Escoffier and many other individuals working on local populations, the degree of spread and how the waves of spreading of polymorphism is quite unique to local populations. And finally, the idea of the thrifty genotype. The thrifty genotype actually hits a whole other component of work in my laboratory on starvation resistance in Drosophila. I won't be talking about this at all, other than to mention that it would be delightful to be able to show Neil this work, because you can recapitulate the whole thrifty genotype phenomenon in Drosophila simply by selecting on starvation resistance. They become outright insulin resistant and sort of in a diabetic-like state in Drosophila. Drosophila do have, after all, an almost gene-for-gene match of homology of genes in the insulin signaling pathway. All right. So his work on inbreeding depression, as I mentioned, was highly epidemiological. You read it today and it's totally unsatisfying. Teaching a course in human genetics, you get to the section on consanguinity and inbreeding, and you realize it's the amount of data on like what really are the medical consequences of inbreeding is fairly thin. There are these sort of statistics of somewhat inflated increase in mental disability and other, the increased rate of pre-reproductive death and so forth. And so it's a problem that's out there that really I think warrants further study. And that further study is coming now because there are large human populations that for many, many generations have practiced fairly high levels of consanguinity. And when that intersects with genomic methods, then we're going to be starting to really make some discoveries. And Joe Gleason, who was at the University of Madison for a number of years, is now at Rockefeller University, was the sort of head of this large consortium known as the Greater Middle East Variome Consortium. The primary focus was schizophrenia, but he managed to pull together collaborators from all across this region of North Africa and into the Middle East, and identify both cases and controls with respect to this particular schizophrenia. He did a total of 1,794 whole exome sequences, and after all their filtering and quality control, winnowed this down to 1,100 high-quality whole exome sequences. At one point, I visited University of Wisconsin-Madison. His student, Eric Scott, who was the sort of heading up the first author on the paper that's coming out on that, pulled me in to sort of help with the population genetic analysis. And we identified very quickly a particularly key problem which had to do with the degree of impact of this level of consanguinity. So basically, about a third of the population marries first cousins and has been doing so since the time of Muhammad, almost 60 generations of that level of inbreeding. And this has had consequences with respect to the regions of homozygosity. So this is in megabase pairs. So African populations, you're looking at about 200 megabase pairs, and it's 2.5 times as much totally homozygous regions of the genome in those populations that have shown this degree of consanguinity. Similarly, you see this in the simple inbreeding coefficient, the proportion of the genome that is autozygous, comparing these African, European, East Asian, essentially, there's some range about 0, but relatively little inbreeding do you see, and yet in these populations, it's on the order of up to 10% or so as an inbreeding coefficient. So there has been an impact of that social custom of marrying first cousins. So this social practice has been going on for 60 generations. The increased homozygosity actually tends to make the population more homozygous. So regions of the genome that are heterozygous, as you inbreed, those heterozygotes sort out into the two homozygous classes. As a result of that shift towards more homozygosity, natural selection for recessive alleles is going to be manifested. It's going to see those recessive alleles now in the homozygous state. And so selection will be much more efficient at removing deleterious recessives in a population that's inbred. And this results in a mutation selection balance that has a much lower frequency at equilibrium for deleterious alleles. And this phenomenon is known as purging. And so purging, one would expect, and we actually did simulations to show that with that level of inbreeding for that long a period, there should be substantial purging, a substantial appearance of reduced frequency of deleterious recessives in a population of that sort. And so we took a careful look at this, and this is the last slide in this little series, which is to compare non-coding, synonymous, non-synonymous, loss of function variants, and so forth among the different populations. So these are in Africa and East Asia. So those are non-inbred populations, and these are the somewhat more inbred populations here. And what you see is that there is a somewhat greater effective population size in Africa, resulting in the greater sort of derived allele frequency in those populations. Whereas in all these other classes, including outright loss of function variants, you do not see this particular tendency for reduced frequency of loss of function variants in those more consanguineous populations. So it's as though the answer to the question, do they show purging? The answer is a clear no. We don't see any evidence that their customs of consanguinity have resulted in a more efficient selection reduction of those deleterious alleles. When you dig into this further and look at individual couples that are highly related to each other so that their offspring are expected to be much more inbred than the rest of the population, you notice that, in fact, the birth interval is lengthened so that there's greater interbirth interval, which is suggesting that they might be suffering from greater loss of pregnancies. And yet, they keep on producing offspring until the family size is roughly matched, regardless of the degree of relatedness between the parents. So this is known as reproductive compensation. It's very well known in avian species, and there's a lot of evolutionary genetics literature written about it. If there is a catastrophe of any sort, the reproductive potential of the individual is to sort of try to bring back up to the sort of normal standard of reproduction for that population. And here the social custom is you should have on the order of X number of offspring. If you have trouble doing it and have to work harder, do so. And that's what they do, and it's basically stopped natural selection against those deleterious alleles. And they're stuck in the same boat as us, roughly the same proportion of the genome that has these deleterious alleles. So that's a fairly interesting result. I think Neil would be impressed that the sort of study of consanguinity is moving forward in this sort of genomics era. This is just scratching the surface. There's a lot more that one can ask about patterns of sharing of IBD and so forth. But anyway, that is going forward. So back to this general problem, and we'll spend several slides now getting back to this issue of complex traits, heritability, and where we are with understanding the sort of genetic architecture of these complex disorders. And let me start with the National Human Genome Research Institute. They're the ones that are spending the big bucks on these problems. And they split it by dividing into different major programs. One of the big programs is the Center for Mendelian Genomics. By all accounts, this has been wildly successful. They've mapped another 1,000 or so Mendelian disorders. These are the sort of disorders that are identified by the genetic counselors, clinical geneticists who identify syndromes. I think this looks sort of familiar. I read about this. There are these other families that have this. They eventually pool together and think this might be the same thing, and then go about trying to identify what are the genes underlying it. Well, as you probably all know, the way we do this nowadays is you just sequence everybody. Sequence the parents, sequence the affected kids, sometimes sequence unaffected kids as well, and simply by computational means try to identify what are the variants that seem particularly aberrant or loss of function variants. And this succeeds about half the time. For about half these clusters of families where this has been done, it's been successful at Baylor and Yale and the other groups that are doing this. And the striking thing I want to point out is virtually all of these have been coding variants or in the immediate coding region of genes. So that's one sort of take home, that it seems like these very large effect mutations that we're seeing in these Mendelian disorders tend to be coding. The flip side of this is the Centers for Common Disease Genomics. And these are the, schizophrenia, cardiovascular disease, these sorts of diseases that are enormous public health concerns, but they're much more difficult to track down genetically. And so we've gone about these primarily by GWAS, assembling large cohorts of individuals. Mapping of these is indirect then. We're just genotyping panels of SNPs that hopefully by indirect connection through linkage disequilibrium have a statistical association with elevated risk. And of the hits, and there are thousands of them, you've all seen the pictures, 98% are non-coding. And this is because 98% of the genome is non-coding, and the method is working by just identifying these sort of indirect associations. And so this might lead you to believe that, okay, 98% of all genetic risk for disease is non-coding, and that would not be exactly the truth here. This is just indicating this sort of statistical association that's identified in GWAS. It hasn't identified what's causal, and we don't really sort of know. In fact, there's enormous ignorance about the following fact is, of these variants, what fraction, well, the fraction of the variants that we explain with these GWAS is very small. This is very well known, this sort of missing heritability problem. But that remaining bit, we all think, okay, there's a certain chance that because the SNPs on the arrays that are being used are common SNPs, maybe rare variants are being missed. And in fact, if there were rare variants in the genome accounting for the differential risk in these diseases being studied this way, they would not be detected by the methods that are being used. That's a key point. So if there were rare variants important, we would not see them. So some fraction of this variance may be many, many different, that is genetic heterogeneity for many different rare variants that may even have fairly substantial effect. They wouldn't be large enough or common enough to be seen in pedigrees, then they would be Mendelian diseases, but there's this sort of middle ground that they could be. All right, so what are we going to do then if we want to know about the genetics of complex disease? How are we going to discover what are the genes, how do they work, what are the variants in the disease, and how do they interact with each other and with environmental exposures? So this environmental exposures is a fairly important thing that's relatively understudied, I think, in much of this area, which is as soon as you look at these sort of quantitative traits in either agricultural systems or in model organisms, the impact of rearing them in different cultures, different temperatures, whatever, different environmental exposures, the fact that the genes that are underlying those traits also interact with the environment is readily apparent. And so how do we understand that in humans where we just simply cannot control the environment very well? So at the end of the day, again, I'll just emphasize, I think what we really want eventually is to get at prediction. And it's a different sort of prediction. It's given the genotype and maybe given measurements of other attributes like environmental exposures, can we predict some kind of prognosis for health? So at the root of all this, I'm sorry I have to give so much history here, but we have to go back and think, like, what is the underlying model here that's being applied so widely in human genetics? And it's really this infinitesimal model. If you're not familiar with this term, there's a terrific review of it by Nick Barton and Etheridge. It's just posted on the bioRxiv. And this actually is the underlying model that Francis Galton and Carl Pearson had in mind when they first developed the idea of relationships between relatives. It was Fisher, after finally we had the sort of rediscovery of Mendel and the sort of incorporation of Mendelian principles in all of the thinking about genetics. Fisher then took this Mendelian sort of paradigm and projected it into this infinitesimal model. What if there are many, many alleles, all that behave like a Mendelian factor that are responsible for traits? Would we get the degree of relationships between relatives that we see? And of course, the answer was yes, beautifully proven in that 1918 paper. However, the infinite alleles model is sort of assuming everything's additive. Alleles are additive within a locus. The effects across loci are also additive. And then we sort of shrink to the limit. The effect size of each locus gets smaller and smaller as the number of loci that are important get bigger and bigger. So that's why it's called infinitesimal. It's many, many, many alleles of very, very small effect. They're all undergoing the normal Mendelian sort of process, but it's a sort of statistical reduction to a fairly strange sounding model that nevertheless has been enormously influential and in fact enormously successful in agriculture. And we'll see that in a second. So from this, you can derive, again, these tools that have been so useful in agriculture, like the Breeder's equation. You can derive things in evolutionary biology about the sort of attributes of mutation, selection, drift balance, and so forth. And again, I refer to that Barton-Etheridge paper. So the other part of the title was heritability. I'm going to start with a very classical idea of heritability, which is, if you think about it, has nothing to do with SNPs or DNA sequence. It's entirely at the level of phenotype. So can we relate the average of the two parents to the mean of the offspring? And the regression to that is this narrow sense heritability. That's how it was seen by Francis Galton. It's the definition of narrow sense heritability to this day. You can derive this in many, many different ways. The Elston-Stewart algorithm, or the Haseman-Elston algorithm is actually also derivable from this infinitesimal model. You get a relationship between proportion of the genome that's shared identical by descent with the degree of phenotypic difference between individuals. The slope of that relationship also is this very same narrow sense heritability under the infinitesimal model, okay? So that's a key kind of caveat. Anyway, it allows prediction of the offspring phenotype given the parent's phenotype. That's very different from what we really want. What we really want in genetics today is prediction of the offspring phenotype from the offspring's genotype, ideally, or perhaps from the parent's genotype as well. But it's a SNP, like sequence-based prediction, which is different from this classical heritability. So why are we so excited about this? Why are so many people applying these sort of agricultural genetic sorts of tools to human genetics? It's because it's been so wildly successful. If you haven't read the Wikipedia page or you don't know this name, you have to go online and look it up. Norman Borlaug is credited with saving 1 billion lives, and this is probably not an exaggeration. The food shortage in India in the early 60s was getting to be just desperate. It led Paul Ehrlich to predict that there would be massive starvation and so forth. And he was pretty much on target if it weren't for Norman Borlaug to come along and through classical breeding methods arrive at the semi-dwarf wheat, which was able to stand up within the monsoon rains of India and result in this, I forget what the magnitude of it was, but this enormous improvement in the yield of wheat in just a very few generations. So classical breeding methods work. It uses all these classical ideas that I mentioned, and that's where we are, thinking, aren't these methods of genetics terrific? So in agriculture, what we want to predict is the mean phenotype of many of the offspring. In other words, we're selecting parents to be the parents for a whole collection in a field, or a whole collection of a herd of offspring. And so we're predicting then this mean of the offspring from the parental attributes or parental phenotypes. This is known as a breeding value. What's the breeding value of the parent that lets us predict the offspring? So one of the methods of doing this that was devised by Carl Roy Henderson, I'll show you a little bit more about him, was called this best linear unbiased prediction. We won't go into the details of that, but basically the idea is that some aspect of the sire will be able to predict the milk yield of many, many, many daughters. And for milk cow breeding, this has been enormously successful also. It's resulted in a tripling of the milk yield of cattle since 1960. Quite amazing response just by applying these tools. And this met the modern era with computers. So it was all devised by this guy, Charles Roy Henderson. His PhD was in animal breeding at Iowa State. He worked with Jay Lush. And what he did was he combined fixed and random effects into a single model. This is this mixed linear model that everybody's so excited about in human genetics. Well, it's not a human genetics thing. It's for cow breeding. We'll get there in a second. So Henderson had a student named Cheryl Searle, a New Zealander, who was terrific at matrix algebra. And Searle was actually the one who introduced the use of matrix algebra in linear models. And the two of them put these two ideas together and came up with this amazingly compact and quite beautiful and elegant representation of this problem of combining the prediction of offspring from the parent's sort of phenotype, and now even from parent SNP genotypes is applicable in that context, along with all the other attributes, like, was it particularly rainy that year? Was this field particularly, low in phosphorus or whatever, these sort of so-called random effects. And so what Henderson did, and this is important history because I'm at Cornell University, he was actually the chairman of animal science at Cornell, and he put the entire New York State dairy herd data on the computer. This was the first use of the largest computer at Cornell. And just by applying this method, it obtained incredibly accurate predictions of the milk yield for successive years. And this was widely adopted then across the entire country now. And again, it has resulted in tripling milk yield since 1960. So again, the point is, I'm not going to downplay the importance of these sorts of methods because they're incredibly effective in this agricultural setting. And so now the challenge is agricultural genetics types have come into human genetics and claimed, let's just apply this to humans, look how well we can do. And the question is this really the direction that we want to go? Just to give you a little bit more familiarity, the idea is you have this vector of fixed effects. So this might be the effect of each SNP on a particular trait. And this will be this matrix of individual by SNP genotypes. So these are sort of fixed effects. And this will be a vector of like the regression coefficients of the effect of each sort of environmental variable, let's say, these other sort of random effects. And this would be then a matrix of those attributes of those random effects for each individual in that particular sample. And you'll notice then that sort of, you can think of it as gene and environment, and there's no interaction, they're completely separate terms and they're just added together. So that's a key thing. It's like It says, forget about any genotype by environment interaction. We're just going to consider this in an agricultural setting where everybody's in one environment at the same time, and we don't need to worry about that stuff. Anyway, Henderson's fame is for deriving a beautiful and elegant and accurate solution to this particular problem. So again, agricultural genetics is predicting the mean yield of a herd, whereas medical genetics, we want to do prognosis for the individual. Again, getting back to that idea of this isn't precision medicine exactly yet, okay? All right, so the application that we're seeing that's quite interesting is this thing called polygenic risk score. Polygenic risk score is basically you do that general linear model, you get your weights for all the SNPs and how they affect each of the phenotypes. You don't bother dealing with which of those regression coefficients are significant or not. You use them all or have some other sort of filtering, but it's not just the significant ones. You use pretty much the genome and then simply add them up. For each SNP genotype, you do the multiplication times that regression coefficient, add them up, and that gives you then a prediction of the phenotype under that model. Now, you think it through, the model is very strictly infinitesimal. It's assuming there's no interaction, no G by E. Every SNP has a small effect, and we're just all going to add them up. And so, to the extent that that model fits, you're happy. Now, the agricultural types say, look how well it's worked for dairy breeding. Why not just apply it for humans, all right? So many papers have been published on this. It was Sean Purcell, 2009, I think was the first paper I could find that actually applied this. And they could explain something like 3% of the risk of schizophrenia, and I thought that was progress. Genetic Epidemiology 2015, Chen et al. Alcus Price has particularly pushed this, done some brilliant work on it. It's very smart. It's like the best work that you could do applying that. What I'm trying to argue is maybe not the best model, this infinitesimal model. It's the model that we have access to. That's their sort of argument. But at any rate, they're integrating things like ancestry. So if you have known degree of relatedness, you can actually tweak the model, get it to fit better. They're still explaining just a few % of the variance. Another one from Alcus Price's group looked at linkage disequilibrium. So in fact, all the SNPs are not independent. The strict infinitesimal model assumes everything's independent. Because they come into the model with a correlation structure, maybe we should weigh that in. That helps a little bit too. And this is a recent paper looking at breast cancer. And what they did in this paper, this was this paper right here, where they looked at quintiles of the polygenic risk score. So for each individual, you have fitted the model. So you have the betas, the regression coefficients. You have all their SNP genotypes. So now, forgetting about the phenotype, just using their SNP genotypes, you can calculate each individual's polygenic risk score. And you can then put them into quintiles. So between, this would be the lowest risk and this would be the highest risk. And then you project those out on this diagram with age, and you can see there actually is an elevated breast cancer risk among the individuals in the highest quintile. So it actually does predict some handful of % of risk. It's better than absolutely nothing, and probably we'll be able to tweak these models and get them to do a little bit better eventually, but there's apparently some degree of improved prediction by these methods. So the real problem has to do with the definition of success here. So under these models, the sort of thing that you're trying to explain is heritability or additive variance. And so Peter Vischer is the key one who's pushing this charge, which is if we can only explain heritability, then we're going to be ahead of the game. And pretty much the whole community is bought into this because the missing heritability problem is seen as being an important problem. There's this missing heritability. Oh my God, we have to figure out what are the genes that are explaining the missing heritability. Only that missing heritability only has meaning in the context of this infinitesimal model, where we're sort of thinking about, everything is this sort of small effect, many, many alleles, and there's some degree of variance that's explained by this genetic variation. So if you set it up where that's the objective, I just want to explain additive variance, then a polygenic risk score is going to be successful at explaining a greater proportion of that variance. But it's of almost no utility for prediction of individual phenotype. And anyway, so this is a kind of a cute paper, if you haven't seen this, when Olchenko and colleagues published in the European Journal of Human Genetics, where they looked at the variance explained by the top 54 SNPs for height. And everybody knows you only explain 4% of the variance in height if you use the top 54, millions of dollars spent on all the SNP genotyping. Whereas if you just look at the parents, you explain 40% of the variance. Kind of funny, but anyway, it's cute. All right, so why are we still trying to predict the milk yield of 1,000 daughters of everybody instead of actually trying to do prediction? Okay, so this model that's trying to fit the infinitesimal model to whole genome data is known as GCTA from Peter Vischer. And it's basically a force fit of that infinitesimal model. A good fit of that model is not proof that the infinitesimal model is true, and it's possible to obtain very, very nice fits of that model. There can be, under the hood, a lot of dominance, a lot of epistasis, a lot of G by E, and yet still those models fit pretty well. And the reason for that is, in any context, every locus will have a marginal effect. You will see those, the effects of the allelic differences at that locus projected to the margin, sum up across all the other loci. Just look at that marginal effect. And then those marginal effects, at some level, you can add them across SNPs. They might not be completely additive, but you do better than nothing doing that. And in the end, you get a fit that's better than nothing, and in fact, highly statistically significant for almost any of these phenotypes. And the reason for this is because you've summed over so many effects of such small size that there's sort of this inevitability, this central limit theorem-like thing that applies that gives us this sort of a fusion of all the effects into looking additive. A way to see this is, so suppose you have many, many pairs of loci that are epistatic, and some are positive, some are negative, some are positive, some are negative. When you collapse over all of those pluses and minuses all sort of wipe out, and you end up with some sort of net effect that in the end you can fit to an additive model. Another way to see this is that even if there's epistasis, a lot of that epistasis will appear to be additive genetic variants if you calculate it in the classical way that animal breeders did. So let's think about epistasis and G by E for just a second. There's a lot of work that's going on in this. It's not very satisfying because many, many people have said, okay, fine, let's look for all pairwise interactions across all SNPs in humans and quantify how much epistasis do we see. And they always fall flat on their face. And the reason for that is there's just it's a dimensionality explosion. If you look at 1 million SNPs, you're looking at 10 to the 12th pairwise combinations of them, and you just have no statistical power to detect epistasis. One exception to this is in cancer, and people have started to think about, if you look at all the SNPs that you see in tumors, and look at the mutations that have arisen in tumors, and identify what are the variants that seem to be drivers of that tumor, and then look at pairs of mutations and ask, when do we see pairs of mutations that are causing those tumors? A kind of funny thing happens is that there's in fact, many pairs of SNPs are present in extreme deficit in the genotypes. So it's as though those pairs of, let's call them loss of function mutations, usually they're fairly large effect, pairs of loss of function result in cell lethality. So this is a classical term, goes way back, Dobzhansky used it in the fly work. Synthetic lethality is where pairs of mutations together result in lethality. And so that's an extreme form of epistasis, right? And so many of these cases of that kind of level of epistasis, you simply don't see. They will die in utero. In this case, the cells disappear from a tumor. You just don't see them when you sequence them. But if you set up the analysis, in this Ryan et al. Review, they sort of talk about this, you see many, many pairs of SNPs that are present in highly significant statistical deficit because of this apparent synthetic lethality. And they're using this then to identify drugs that knock out those pairs of pathways, and combinations of drugs then exert this synthetic lethal effect. And it's extremely promising in the cancer field. And that's just showing it. Okay, for G by E, genotype by environment interaction, you need some consistent, relatively high impact phenotype to make progress here. And there are many papers that you're starting to see on smoking. So smokers versus non-smokers, massive effects on gene expression, particularly in the lung epithelium of smokers compared to non-smokers. If you ask in them, are there pairs of SNPs that impact gene expression? So EQTLs now, do you see pairs that impact expression in a way that departs from additivity. And there are many, many instances of very strong epistatic interactions between pairs of EQTLs in smokers versus non-smokers. And so this is, again, a G by E effect. Okay. Okay, this is just sort of the sum of these effects. Epistasis and G by E may contribute little to the additive variance, but they contribute a lot to departures of individuals from predictions of purely additive models. And to get at this, we actually did some simulations. And these are going to take just a second to explain, but you can generate a circumstance where there are particular levels of variance. So you hardwire particular patterns of expression where there's dominance and epistasis or interaction here, and certain additive and environmental effects as well. So we hardwire a 10 locus model with these, and then we calculate what's the relationship between the phenotype of the midparent and the offspring for genotypes and phenotypes that we generate from this model, all right? And when we do that, this is a cooked example where we end up with a heritability of 0.8. Well, with this degree of interaction and dominance, we can end up with a situation where the pattern of epistasis, despite this level of additive variance, this degree of heritability, you can end up with pairwise interactions that result in quite poor performance of this prediction, of individual prediction. So this is where you predict everybody's phenotype from all the SNP genotype data, leaving one out, and then add that individual back in and calculate what's the observed phenotype versus the predicted phenotype. Relatively poor correlation, in this case, just 0.09. So this is, again, this emphasis that on the one hand, these models can predict a population phenomenon at the level of individual prediction from SNP genotype data. They can do relatively poorly. The converse, amazingly, is also true. You can have models where there's relatively little additive variance, relatively poor sort of parent-offspring regression, heritability here of 0.4, and yet a much, much stronger ability to predict, simply because there are enough rare large effect alleles where the prediction of those individuals is quite strong. So again, the emphasis here of the take-home from these simulations is just there is a pretty strong decoupling between the sort of thing that's going on with the infinitesimal model, heritability, the polygenic risk score kind of approach, versus if you actually know the hardwiring of individual genes, the prediction of individual phenotype from genotype can be fairly well decoupled from that polygenic risk score. All right, how much time do we have? 10 minutes? 5 minutes? Okay, so I think I can do this in maybe 5 or 10 minutes. So I'm shifting gears radically because this is one radical disagreement with James Neal. So put that on the record, okay? So he didn't like flies at the end. He thought, okay, we've done everything we need to do with flies. And at the end of the book, he actually says model organisms in general have had their day. Let's just work on humans. Almost that strongly, maybe not quite. And many people have this attitude. In fact, the funding for Drosophila research at the National Institutes of Health has fallen precipitously over the last few years. And so I can give this story simply because it's an alum from your illustrious institution. Clement Chow was a student here, worked with Mary Meisler, and he did a postdoctoral fellowship with me and did a terrific study that I just like to advertise for him to show how wonderful it is. And it's on retinitis pigmentosa. This, of course, is a progressive loss of vision due to loss of rods end cones. It's a degeneration of the neurons, the most common form of retinal degeneration with a birth incidence of about 1 in 4,000. There's an autosomal dominant retinitis pigmentosa that makes up about 30 to 40% of the cases. So you're all familiar with RP. One form is a defect in rhodopsin. So particular missense changes in rhodopsin, you can think of them as eliciting an unfolded protein response in the retina, which then results in the apoptosis of the retina, which results in RP. So dominant mutations comprise about 25% of the cases. It's a particularly exciting looking protein. It's a GPCR found primarily in rod photoreceptors of the retina, and it's this particular common mutation does cause this misfolding and induction of the unfolded protein response, which can result in cell death, as I mentioned. Okay, you can actually make this particular mutation. It's a rhodopsin. Flies have rhodopsin. They can see. They express rhodopsin only in the eye. And this change from glycine to aspartic acid does actually result in misfolding of rhodopsin. And that misfolding, if you express it in the eye, results in this nasty phenotype. So the eye facets fuse, it gets much smaller in size, and it's because of cell death of cells within those ommatidia of the eye. So you can use this as a phenotype for to what degree am I seeing cell death in the eye. And we can then quantify this. And this paper, Kang et al. 1912, actually, or 2012, developed this particular variant. So Clement's big idea was to use the particular construct and use the natural variation across the whole genome to identify enhancers and suppressors of this phenotype. So here's the basic idea. With the promoter from the glass, this glass module reporter, GMR, that can drive GAL4, a yeast transcription factor, to be expressed only in the eye. When that's expressed in the eye, the GAL4 transcription factor causes the UAS promoter to drive expression of this rhodopsin defect in the eye. So this means that we're not getting this nasty protein expressed all over the body of the fly, which would kill it. It's only expressed in the eye where we can report the phenotype. We recross that. This is the DGRP, or the Drosophila Genetic Reference Panel. So it's a set of, what we did it with was 174 of those fly lines. So we take virgin females of this one stock, and we just collect males of the 174 lines. And in 174 separate vials, we just set up the matings. You can do that in an afternoon. And then the F1 hybrids then are going to be heterozygous for this construct. They will express the rhodopsin in the eye, even though they're just heterozygous for it. They express it at a level that does cause the eye degeneration. And the other whole part of the genome is from DGRP. So any other alleles from the DGRP line that differ from one line to another will now be heterozygous across the whole genome of these F1 progeny. So these F1 progeny actually have an eye phenotype, and it turns out to be variable from one line to another because of the genetic backgrounds being variable across these DGRP lines. So all we do is take images of the fly eyes. It's easy to just pop the flies in a freezer and then take photographs of the eye and with a computer measure the eye size very efficiently over the next couple of weeks. And then we identify genes by doing basically a GWAS analysis because we have whole genome sequence of all these lines that we've crossed these flies to. This is showing a distribution of the eye size across the lines just in rank order by their means, and you can see that there's a terrific and highly significant difference in the magnitude of their eye sizes. And this is just to show you can see it very plainly with the naked eye. In fact, you can even see the degree of necrosis that's sort of correlated with eye size. It's not perfectly correlated, but this eye has relatively little visible necrosis, and you can see this kind of nastiness on the eye that's from the actual cell death that's occurring there. So this pulls up lots of interesting genes, and the exciting news is that Clement, who's now in the Department of Human Genetics at Utah, is collaborating with human geneticists there to track down several of these candidates. Some of these candidates are particularly important for this particular phenotype. If you look at GO terms, these gene ontology terms, several of them are involved in aspects of eye development, which makes sense. But in addition, 50% of them are associated with some form of retinal disease in humans. So the homologs of these fly genes have actually been implicated in retinal disease in humans. So some of these you'll perhaps recognize. So here's one, Bardet-Biedl syndrome number 9. Bardet-Biedl syndrome is a ciliopathy often associated with retinitis pigmentosa, for instance. And if we then do the next experiment that's fairly easy to do in flies, knock out those single genes with RNAi and ask, do they have an effect on the phenotype? Here's the control. So if we knock out any of these genes here, it results in a suppression of or decrease in eye size. In other words, it's enhancing the effect, resulting in a more severe phenotype. Not all of these candidates had an effect, so there were a few that are marginal and a few that really didn't seem to have any effect, and then a few that are actually suppressors of the effect. So this is giving rise to lots of hypotheses about how are these genes interacting, what are the pathways, how can we track these down in actual human patients as well. And just to show the specific examples of just a couple, these RNAi experiments really show this fairly large phenotypic effect when you knock these genes out. Here's the one that's Bardet-Biedl syndrome again. All right. So the conclusions from this retinitis pigmentosa study are that these modifier genes result in a wide range of phenotype across these different fly lines. We can screen them in a very efficient way. Setting that up, as I mentioned, just takes less than a week. You can sort out the flies in less than a week. And then it's a week or two to take the images. We could probably have automated that even further. We're pursuing this now for amyotrophic lateral sclerosis. There's a particular variant of that also you can express in the fly. It results in eye degeneration. And nobody has a clue about what might be the suppressors and enhancers of that particular phenotype in the C9orf72 form of ALS in humans. So we're pursuing that with Nancy Bonini at UPenn. So Clement likes to think that this idea of Mendelian diseases, there are many different Mendelian diseases that actually show quite variable penetrance. There are individuals homozygous for beta-S globin who have shown no symptom of sickle cell. If we then take this sort of to a continuum, the rest of the genome can be thought of as screening for these suppressors and enhancers of that phenotype. So this idea of variable penetrant shouldn't be thought of as a nuisance. It's actually a gold mine that can be utilized if we have the right experimental design to track them down. So that's my plug for flies. We can talk about that over drinks later. So the overall conclusions, this is the end. Infinitesimal model has been of terrific value in agriculture. So cheer it on. It has guided progress in classical marker-assisted breeding. So using SNPs to actually incorporate that into those models is terrifically efficient now for all kinds of breeding programs and all kinds of very non-model crops, for instance. The limits to the wholesale application of these animal breeding methods in human genetics, polygenic risk score is likely to have relatively limited clinical application. That's my own personal view. Improvements over PRS will, however, require understanding of two key aspects of this sort of context dependence. And these include epistasis and G by E. And I'm delighted to be able to say that in front of Charlie Singh, who was a great mentor in thinking about these sorts of problems. So as these individual prediction methods improve, maybe James Neal's sharp division between the analysis of the gene pool and clinical care for individuals will fade away. That is, we'll get to the point where by thinking about the gene pool and thinking about it at this sort of population level, we'll be able to apply these statistical epidemiological sorts of methods, get back to the actual mechanism, and then actually be able to use that for some sort of individual guidance of either drugs or changes in behavior or changes in diet or whatever. Okay, nobody does science by themselves anymore, so I've been blessed with a terrifically effective lab group, a fun group to work with, and leaving this up for a second for you to play Who's Waldo to see the three people that are actually hail from this very university, and here they are. So there's Clement Chow, did that fly work I just talked about. Chris Van Hout was a student with Julie Douglas and is now at Regeneron doing terrific work there. Tim Canalan and I published 12 papers during his postdoc. He's a student of Lacey Knowles in the biology department here, and Tim is now at Monash University doing great work there. The interest in the pure kind of theory work that he does seems to be waning here. That's even more shocking to me given how terrifically productive he was, but they took him with open arms at Australia, and he's doing terrifically well. Tricia actually got her undergraduate degree here, was a graduate student with Sean Carroll at University of Wisconsin. And she, of course, did a postdoc with me where we did a lot of work on allele-specific expression, and now she's back here in your biology department. And sorry she couldn't be here, but anyway, thanks very much for your attention, and that's it. Are there questions, or that's it? Yes.
There's a question from the Twitter audience. I quoted it where you said the sum of many, many small marginal effects looks additive. And they were asking if the marginal was well defined.
No, I didn't really define it at all. No. So marginal with respect to, collapsing over the effects of all the other loci. That's all that I mean in the statistical sense of marginal. Yeah. Charlie?
Stuart Coughlin and Andreas Wagner have come up with an idea that Stuart Kaufman has a new book out
I
on,
haven't seen it.
yeah, on his recombinant ideas in biology and sociology and politics and everything else. And, and you know, Andreas's book, The Emergence of recently. But the idea is that there's, with so many possibilities in the genome and so many possibilities in the environment, that every point in time is just one small realization of all the possibilities. And we've got a shifting or a moving population of inference. We really don't have a handle on the gene pool. We don't have a handle on all the environmental possibilities, especially if it's changing over time. So if you think of the whole universe of possibilities that could happen in biology and genetics, we're only seeing a very small part of that universe at any one time. Now, this may not be true, although you point out with retinal, in this last example, may not be true for the single gene Mendelian traits, but it has a little bit of it because all of these possibilities probably aren't realized, all these modifiers in any one, what I call population of inference. So what are we gonna do with this idea about what is the population of inference? And it's not being realized, or you know, we're not representatively sampling it at any one time. We're just seeing little patches of it.
Yeah, so this idea that, yeah, you're only seeing a sort of a collapse of dimensions of reality, and there are other sort of causal factors that you never mention, the sort of Simpson's paradox kind of problem of unobserved variables impacting what you're seeing. I mean, this is always going to be the case, and you're a great proponent of despair for these sorts of problems. But I think what I would say is that there are some Mendelian factors where we have very good prediction for those. And so it's like, how much further can we go? Surely there will be better abilities to predict for relatively larger effect and then sort of moderate effect, and then it is going to fall apart. And I agree with you completely that at some level we have to admit there is sufficient complexity to this problem where our prediction's never going to be perfect. In fact, an awful lot of the time it cannot be much better than kind of, you know, meager, but there will be more cases where it will be better than it is today. I'm just convinced that's got to be true. Now, if you were to ask, so how do we understand about those unknown variables? Well, we don't. We're still having to work within this kind of collapse of the dimensions that we actually see.
I think there's a part of the community, in the scientific community, that's getting concerned about our inability to predict. And I think we're at risk for not realizing some of the larger issues and not dealing with them.
Yeah, no, I mean, the overselling of, and that's why I think prediction hasn't been targeted as this is what we want to do. I think by my emphasizing this is at the end what we really need to do, it focuses on why is this so hard? Because people are sort of hiding from that problem. And we certainly do not want to oversell it. It will be hard. It will continue to be hard.
We're avoiding a lot of inconvenient truths.
Yeah.
So a societal question, how do we counter public perceptions, perhaps over-perceptions of genetic determinism?
Yeah. Yeah, So, I mean, there's been great ignorance on this issue, right? So I think it's a matter of just gentle persuasion. People get these ideas for, you know, they're a product of their environment and so forth, and get to these ideas from fairly uninformed kind of background about these notions. And so it just takes education. It's going to take care and patience to kind of step through what do we mean by these phenotypes? What is the impact of genetics actually on such attributes as intelligence and so forth? And why is it so difficult to disentangle social, cultural, environmental kind of things from the genetics for such complicated traits. I don't think there's anything much to say other than it's just going to take, concerted care and education. I suppose you could also sort of wail at the press for just trying to feed into controversy and not being very good at helping to educate the population about scientific issues. You could wail at some politicians for being stupid on the issue as well, but it's really, I mean, this community, the American Society of Human Genetics, I think is doing a terrific job with respect to their perception of the importance of public education. I think they really do make an effort, but we could all do more. I mean, we all sort of bear a responsibility to do our part to help in that education process.
I have a much simpler question.
Good.
So you're contrasting the predictability of, let's say, height with 52 or so top SNPs from height, or even with PRS, you get maybe 15%, and then simple parental phenotype predicts 40% of the phenotype. Do you know how much of that might be actual environment? Because parents that feed their children a certain diet, parents who are aggressive, exert this effect on their children. So parents usually give not just the SNPs, but also the environment on it. So is it possible that a good share of that 40% of the parental predicting phenotype in the next generation is involving also environmental or gene times environmental factors?
So that's an excellent question. And one of the arguments in the whole missing heritability world is that, well, we've been overestimating heritability because of things like shared environment. And there actually is interest in sort of improving estimates of heritability by using, for instance, large pedigrees, like in the DECODE project. I think Augie Kong has a paper that's coming out where people have been re-estimating things like heritability even for height. And the typical number you see is like 80% is the sort of classic heritability. And it is coming down a bit. They're talking more like 60% instead of 80%. So undoubtedly, there's some shared environment effects. Some of those designs try to explicitly even quantify it. That's not easy to do. Designs like with the offspring of identical twins, being half siblings, these sorts of designs to some extent can quantify it. But you're right, it will come down some, and you'll see a few papers coming out in the next couple of months that are these sort of updates of basically classical heritability that take a much more careful look at shared environment.
Okay, well, if there are no more questions, another round of thanks and applause. And please join us, take a look at the posters that are available outside in the atrium.
You did good.
I stayed the whole time. It made me nervous. Shouldn't we be going?
Oh, dear. Promise me we can reconnect. You have some time to talk. But without Debbie, I don't know.
Thank you very much. We'll actually collect you and get you to dinner, but you can relax a little bit, or you can go out and there's some beverages and things available out there. We'll see you tonight at dinner.
Yes, you did. Now it's picture time.
Oh, even better.
Yeah, without